Hi, my name is Rubén García Domínguez, and I'm honored to have this opportunity to present my research at the ISMB Congress. Today, I will walk you through the key findings of my study, which is called Unraveling the Contribution of Three Distinct Paths of Protein Evolution Across the Tree of Life. So let's begin. Let's begin with the introduction. Proteins are composed of domains, which are modular units conserved across multiple sequences. They play a crucial role in protein function. By analyzing protein domains, we can gain valuable insights into the evolutionary processes that shape protein architecture and understand their functional significance. One of the ways to define domains is as blocks in multiple sequence alignments, representing conserved regions across related protein sequences. This is the PFAN database definition that we will use in this study. Protein architecture is defined as the linear arrangements of domains. They can occur as single isolated domains or in combination with other domains, forming intricate architectures. Understanding the distribution and arrangement of domains within proteins is essential for unraveling their functional and evolutionary significance. Protein evolution involves three distinct paths. First, the creation of new domains or divergence of existing ones. Second, the recombination of existing domains. And third, the repetition of existing domains leading to tandem repeat proteins. The occurrence of repeat architecture is known to correlate with organism complexity. Our objective is to evaluate the contribution of these three different paths of protein evolution in different proteomes. A detailed global analysis of this path of protein evolution in different phyla has not been realized yet. Now let's continue with the methodology. First, the dataset selection. To carry out our analysis, we obtained a comprehensive dataset of around 21,000 proteomes from the UniProt reference proteomes database. Viruses were excluded and proteomes were filtered by completeness. To ensure a balanced representation, we implemented a resampling approach and randomly selected a maximum of 101 proteomes per clade. Phyla with fewer than 10 proteomes were excluded from the analysis. This rigorous selection process resulted in a final dataset of 1,212 proteomes. Using the PFAN-SCAM script from the PFAN database, we identified domains and clans within each proteome. We then reconstructed domain architecture for each protein based on the presence of significant domains. Quantitative analysis involved measuring the number of domains and domain pairs to assess domain diversity and combination, as well as the full architectures. Consideration of domains as repeat was based on PFAN annotation. The recombination rate was calculated as the number of domain pairs divided by the number of domains. Additionally, the proportion of repeat architecture was calculated as the proportion of the total architecture that contains at least one domain annotated as repeat. To analyze the data and identify significant differences, we perform various statistical tests that allow us to compare different groups and detect patterns and variation in the data. So now let's move on to the results. We conducted a quantitative analysis to assess domain diversity, domain combination, and the recombination rates across different phyla. Here are the key findings. Our analysis revealed a strong divergence in domain identity, particularly in Eukaryota. However, certain phyla from bacteria, such as cyanobacteria and protobacteria, have also undergone significant domain divergence and/or novel domain emergence. Also, we observe variations in the number of domain pairs and recombination rate across different phyla, indicating diverse mechanisms of domain rearrangement. Some bacteria phyla, as Plantomycetota and the Sulfobacterota, show high recombination rate, even higher than some eukaryotes, like fungi. Next, let's delve into the analysis of total architecture and the proportion of repeat architectures. We found that certain phyla, like Plantomycetota and Cyanobacteria, exhibited distinctive patterns in terms of repeat architecture compared to other phyla, even overlapping again with fungi. In Eukaryota, especially Chordata, we observed the highest proportion of repeat architectures. These findings highlight the importance of repeat architecture in driving complexity in evolution. As conclusions, clear tendencies of evolution can be observed through both divergence of domains identity and recombination of domains. Eukaryota exhibits the highest levels of divergence in all aspects, including domain identity, recombination rates, and proportion of repeat architectures. Archaea, despite being more closely related to Eukaryota, display the least divergence in all aspects. We could say that they are the least complex organisms. Plantomycetota, although not exhibiting a significant divergence of domain identity, has demonstrated a higher recombination rate and proportion of repeat architectures, which warrant further investigation and challenge the generalization that bacteria are inherently simpler. I would like to express my gratitude to my research team and funding agencies to their support in carrying out this study. Their contributions were instrumental in achieving the results presented today. So thank you all for your attention, and I'm open to any question you may have.